Scientists solve mystery of sea star mass die-off. What killed 5 billion sea stars? It's one of the largest marine die-offs ever documented, affecting more than 5 billion sea stars almost overnight. And most people still don't realize just how high the stakes really are. Scientists have finally uncovered the culprit behind this massive die-off, which first caught the world's attention in 2013. Sea stars, often referred to as starfish, usually come in a rainbow of colors and can have anywhere from 5 to 24 arms. They play a crucial role in maintaining balance along the Pacific coast, but a mysterious disease began to ravage them from Mexico all the way to Alaska, leading to a decade-long epidemic that continues to challenge researchers today. But here's where it gets controversial. For years, starfish turning into goo solved. Bacteria behind zombie outbreak revealed. Imagine witnessing a scene that feels straight out of a horror film. A starfish starts twisting, its arms contorting into grotesque shapes, followed by each limb detaching and making its own gruesome journey, all while the creature's skin erupts with sores and its organs ooze out. Eventually, this once vibrant sea inhabitant is reduced to a soupy puddle of viscous goo. This shocking transformation has recurred billions of times along the Pacific coast over the last decade, as a mysterious wasting disease has laid waste to approximately 20 different species of starfish, often referred to as sea stars, stretching from Alaska all the way to Baja California in Mexico. The outbreak, which started in 2013, has left scientists puzzled as to its origins. However, a promising breakthrough was announced by a group of researchers recently. They have finally identified a potential culprit, a bacterial relative of the pathogen that causes cholera. For a long time, I have pondered this and felt an unsettling sense of mystery surrounding the agent responsible for this devastation, remarked Drew Harvell, a researcher affiliated with Cornell University and the University of Washington, who was a co-author on a paper published in Nature Ecology Evolution, detailing this significant discovery. Having a definitive answer at last is incredibly gratifying for me, especially after all this time. Understanding the cause of this wasting syndrome is paramount for conservation initiatives, including efforts to breed a resilient strain of starfish that can withstand the disease and to rejuvenate the ecosystems where they thrive. The dwindling starfish population has led to an explosion of the sea urchin population, creatures that starfish typically prey upon. In turn, this unchecked surge of urchins has ravaged kelp forests, vital for capturing carbon and preventing coastal erosion. Jason Hodin, a senior research scientist at the University of Washington, who also co-authored the study, remarked, "Our lack of understanding regarding the disease has really impeded our ability to implement various restoration strategies effectively. However, not everyone in the starfish research community is completely convinced that the strain of Vibrio pectinicida bacteria linked to this study is indeed the primary reason behind the die-off, which is marked as the most extensive marine epidemic documented in the wild. Researchers have made incorrect connections in the past, such as mistakenly blaming a virus for the wasting disease. Cornell marine biologist Ian Hewson emphasized, "It is essential not to rush to conclusions." In their latest study, researchers zeroed in on the sunflower sea star, a formidable predator capable of reaching the width of a hula hoop and feasting on clams and sea urchins with its numerous limbs, sometimes as many as 24. Since the onset of this outbreak, it is estimated that over 5 billion of these sea stars succumbed to the wasting disease. By 2023, the crisis had escalated to a point where federal authorities proposed to list the species under the Endangered Species Act due to their alarming decline. The research, which unfolded over a 4-year period, demonstrated that healthy starfish would perish when exposed to untreated fluids drawn from afflicted starfish, while those that were exposed to heated fluids remained unharmed. This pointed towards a microbial origin for the disease. Genetic investigations of the deceased starfish began revealing hints that a strain of Vibrio bacteria was responsible for the tragic fate of the sunflower sea stars. This bacteria shares genetic ties to the pathogens that trigger cholera and various foodborne illnesses. Furthermore, the researchers uncovered similar genetic markers of this bacteria in wild sunflower sea stars deteriorating off the coasts of British Columbia and Alaska. The most compelling evidence emerged when the team successfully isolated and cultivated a pure culture of the Vibrio bacteria. During experiments in the laboratory, the vast majority of sunflower sea stars exposed to this specific bacterial strain showed signs of melting away, while control specimens remained healthy. Melanie Prentice, an evolutionary ecologist affiliated with the Hakai Institute and the University of British Columbia, who headed the research, shared that her team spent countless years attempting to debunk their theory about the bacteria's role in the wasting. Ultimately, we had to conclude, okay, we might actually be correct here. But it wasn't without its challenges. Hewson, who did not collaborate on this particular study, highlighted the importance of being cautious about overinterpreting these findings. He pointed out that the team focused their experiments on just one species of starfish, suggesting that other species affected by similar wasting conditions might have succumbed to either different pathogens or varying environmental stresses. Furthermore, injecting starfish with bacteria, something the research team did in some experiments, might provoke a response that wouldn't necessarily align with real-world scenarios. The gruesome disintegration process may in fact be triggered by several different stresses too. Carrying out such experiments is quite complex, and correctly interpreting the outcomes can be quite challenging, he added. Back in 2014, Hewson, along with Harvell and others, published a study attributing the large-scale mortality of starfish to a virus known as a densovirus. However, subsequent trials by some of the same researchers were unable to replicate those initial results. This time around, Alyssa Lois-Geeman, a marine disease ecologist also affiliated with the Hakai Institute and the University of British Columbia and a co-author of this new study, feels confident in their conclusions, primarily because the team successfully cultured the Vibrio bacteria outside of the starfish, allowing them to reinfect healthy specimens. In the previous investigations, researchers had failed to cultivate a pure densovirus culture, which prevented them from completing this crucial step. Throughout their experiments, they also demonstrated that the disease could spread not only through direct injections, but also via mere contact with shared water. We were well aware that we were working in a field where previous answers had proven inaccurate, so we approached our investigations with extreme caution. Eamon said, "We dedicated ample time to confirming every aspect of our findings. Each experiment was repeated, and we did not rely solely on a single trial for our conclusions. With this newfound understanding in hand, scientists are now keen to explore the potential reservoirs of Vibrio bacteria in the vast ocean, run laboratory tests on other starfish species to observe their responses to the pathogenic threat, and investigate whether rising water temperatures could be a factor in triggering these outbreaks. The overarching aim is to cultivate sunflower sea stars that are robust enough to withstand the wasting disease, and subsequently use them to boost the populations across the Pacific." As Harvell concluded, this discovery is a monumental step towards the success of these restoration efforts, without knowing what is causing the deaths. Our ability to tackle the issue would have been severely limited. This situation raises several compelling questions. Does our understanding of marine ecosystems dictate how we should respond to such crises? And how might conservation efforts evolve in light of these findings? I'm eager to hear your thoughts. Do you think the link between Vibrio bacteria and the starfish die-off is conclusive, or is it too early to celebrate a breakthrough? Let's discuss.